Thank you for the opportunity to present what we've been doing at HARC and with collaboration with a lot of other people, especially researchers at PBARC. I'm going to talk about our breeding effort, and mostly it's focused on breeding for coffee leaf rust resistance. Okay, this project is funded by USDA SCRI grant. These are the people by the way, I show a lot of these slides in past talk before, so I might go through it fairly fast, but you're welcome to interrupt me anytime if you have any questions. So these are the project investigator involved. HARC's breeding program started with Dr. Chifumi Nagai in the 90s. Yeah. And she had a foresight during that time. She had a foresight to import coffee leaf rust resistant cultivars into Hawaii. That's Katimor, and she already made the cross between Katimor and all the different variety that's already grown in Hawaii at the time. And the original cross was made in 1999. We had the F1 population sitting in our field, but because we did not have coffee leaf rust, the project just stalled. In 2020, the first case of COR were detected on Maui, and we applied for a new grant, and not only to continue the Katimor crosses, and also we want to also introduce another resistant gene from Obata. Obata and Katimor carry slightly different resistant gene. They are resistant to different panel of the COR races. As well, we know we only have one race in Hawaii so far that we know of, but there are more than 50 around the world. And Katimor resists to the current one we have, it may or may not resist to the future one that we're going to get eventually. And normally in a breeding project, we take 5 or 6 generation to produce a new variety, a new cultivar. But if we do that with coffee, we'll always be playing catch up. So the idea for this project is to see if we can select in the F1 or F2 population and stop there. However, this may involve single tree selection. So once you select some trees, you still need to propagate it and do larger trial in order to be confident to release the cultivar. Okay, so Chifumi and I are responsible for generating the progenies with the resistant cultivars. And then Lisa Keys, Dr. Lisa Keys at PBARC is responsible for the bioassay. And Qingyi and Dapeng were doing the DNA marker to confirm their genetic identity. Also, Qingyi is mapping the resistant gene. And then the field trial originally was planned to be done in Greenwell in Kona, on Greenwell Farm in Kona, Kanapali Coffee Service on Maui, Doe on Oahu, or Wailoa Coffee on Oahu, and Kauai Coffee. And I'll explain a little later, we don't have a trial on Kauai. And Tracy will be involved in phenotypic data collection and including cupping and whatever information generated, Andrea will be responsible to disseminate it. I guess this is what we're doing right now. Oops. Okay. So in 1999, Shifumi made many cross between, can you see my cursor? T5175 and TA667 are the two selection of Katimor we have. And she crossed with, for example, MA10 is a red Katrai originally found on Maui. KO34 is a Typica found in Kona. KO32 was originally from Greenwell's farm. KA17 and 18 are yellow catrai from Kauai. MA2-7 is a mocha type originally found on Maui. Anyway, she made lots of crosses between Katimoa and these cultivar that's already grown in the islands. And she generated F1, and we selected F1 and generate the next generation, F2. And this population, we actually had F2 growing in the field, but the reason is for genetic mapping purpose, and that's the population Qingyi is using to trying to identify the resistant genes. So I'm trying to mimic what Chifumi did, but with Obata. And these are the crosses we made with similar variety. Also, we have some Ethiopian accession at HARC. They are nematode resistant. So we're starting to try to cross with Obata. So there will be, hopefully there will be both nematode and COR resistant. I submitted a different proposal to expand this portion of the research, except it didn't get funded. But we already made more crosses than these. We will continue the project. Okay, so all the seedling we generated 3,000 seedling with all the control plant. At one time I had 5,000 coffee seedling growing in the greenhouse. And we need to screen the progenies with bioassay to identify which one is resistant, which one is susceptible. So in Lisa's lab, they use the leaf disc assay. And in our lab, instead of taking the time to make individual disc, we just cut a piece of leaf tissue, and instead of using 16 leaf discs, we drop 16 drops of the spore suspension on the leaf. And as you can see, the susceptible plant, about 3-4 weeks later, we start to see the spore. Can you see that? Okay. And start to showing symptom. This is a close-up look. We call this susceptible, and we call somehow the tissue will produce these spots but never produce spore. We call that tolerant, and some has no spots at all. And if you wonder why we cut our leaf into this dovetail shape, it's because we notice when the leaves are getting old, it starts to decay from about 1/3, no, on the midrib, about 1/3 towards the petiole. That's why we always cut this part of the midrib off. We also notice the symptom at the bottom, closer to, in the leaf blade, closer to the petiole, the symptoms showing up faster. So we didn't want to just take the top half of the leaf and miss this part. Anyway, we screened through thousands of plants this way, but we also compared the two different methods, the two different labs from Lisa's lab and our lab. There's about 10% differences. So we know either method is not 100%, but we can live with 10%. Okay. I have 2 graduate students that's struggling with the bioassay in the lab. So apparently not everybody can do it. Now, overall, if we look at the ratio between, if we lump the resistant tolerant plants together, the ratio between resistant to susceptible, it's overall about 9-7. 9 resistant, 7 susceptible. If you break it down, it's equal to 9-3 to 3-1. And that fits pretty nicely with the Mendelian system of two dominant gene required, and both are required at the same time in order to be uh, resistant. Uh, and we're kind of happy with this result because I think Qingyi also mapped the gene into two region of the chromosome. So we're pretty happy with that, and I can't wait for Qingyi to find exactly which gene it is. So for all the ones that passed the bioassay, we also did genotyping with 96 DNA marker to verify they are what we think they are. And indeed, for the Katimora F2s, they are all what we think they are. But for the Obata crosses, we did find three crosses. It's actually not cross at all. It was just the selfing of the female parent, and a fourth crosses. It's a mixture. So it was my first time trying to make crosses. Overall, I'm still happy with the result. So next thing is now we generated coffee seedling, screened them for resistance or tolerance, and we verified their genetic background. So it's time for the field trial. So at Greenwell Farm, the plants were planted April a year ago. By the way, I didn't know when I signed up for this project, I have to run logistic because to ship 400 plants in a pot to Outer Island take a lot of coordination. Maui field trial was installed March last year. Doe was the first one actually in December 2024. However, during this time, I applied for shipping permit with HDOA at the same time with Kona and Maui. HDOA did not provide me with a permit because they claim at that time there's no CBB on Kauai, and which we know it wasn't true, and we're trying to change the rule. Apparently, they agreed to change the rule. However, the rule was not finalized. So to this date, I have not getting a permit. And on top of that, there was the lease issue about the fate of Kauai coffee. So we decide not to ship plants to Kauai, and we're planting those seedling that's set aside for Kauai coffee at Kunia Farm at HARC. In addition to the four original field trial, each of these field trial, by the way, has 300 either resistant or tolerant plant from the Obata or Katimora crosses, and then has 100 control plant. The 100 control plant, including 60 is susceptible and 40 that's resistant. So we have XX plants. So I shipped it to Tracy, and it was planted last June. And also we have a demonstration site to farm under the solar panel on Oahu in Mililani. I'll show you a picture later. And we also install a smaller trial under the solar panel. We're in the process of looking at their field performances, and eventually we want to check the cupping quality using metabolomic analysis to predict to reduce the number had to cup. One other reason we don't recommend using Katimora directly as a cultivar for farmer here in Hawaii. It's because when they came out with the Katimora cultivar, all the focus was on CR resistance and yield. They did not take into cupping quality into consideration. And we also don't know how Obata and Katimora would do in our environment. That's why we made the effort of crossing them into the cultivar that we already grown here. Okay, here's a picture of the field at Greenwell Farm. This picture was taken in January. At that time, about we found one plant that's infected with COR, and which it was a typical control plant. Thank goodness. I'm sure there's more now. There's more plants now. And I was told during the Kona low, this field had about 10% damage. But okay, this is a picture of Kanapali coffee taken in February. About 80% of the plants have flowers on them. So we're hoping this year we can do at least some harvest and check the quality. Uh, in February, we did not see any COR, any real COR symptom. I'm sure there are some now after all the rain we got. Uh, unless there's another reason, my next visit to Maui would be in July. This is the first field we installed in Wailoa Coffee. About as of this month, about 90% of the plants are flowering, and we found 75% of the susceptible control plant are showing COR symptom, and about 3% of the plant that passed the bile acid also showing symptoms that represent about 8 out of 292 plants. As I mentioned earlier, the two we know from comparing the two lab result, bile acid result, we know our effort is not 100%. However, in compared to susceptible control, we're pretty happy that we actually eliminated most of the susceptible plants. Okay, and these susceptible field-grown susceptible plants actually can provide Qingyi with material to verify the resistant gene when they find it. This is a picture from Tracy. This is what they look like in at Pig Bar, but it was raining cats and dogs, so I don't have any data. This is the field trial under the solar panel in Mililani. This facility is called Hawaii Agrivoltaic Research and Demonstration Center. It's run by my colleague Julie Burden, and this is her email. If you're interested in, she grows all kinds of crops under the solar panel, all different vegetables. She grow very, very good strawberry. She also do bushes or ornamental plants or mamaki tea. And she even trying to grow mushroom from out of albizia or African tulip under the solar panel. And we also, so this is what the plant look like right now. There's no flower yet. The idea is to show, because most of the land that's good for solar farm, it's on ag land, but we don't have to take farming. We don't have to, take ag land out just to produce energy. We can do both. Okay, so as I mentioned earlier, especially in the F2 population, every single plant is considered different. So our selection will be based on single tree selection. And we will have to have way to propagate it vegetatively to increase the number of plants to do further trial. So one other way is using tissue culture through embryogenesis, somatic embryogenesis. And right now, we're also part of a different SCRI project producing selected coffee cultivars. So right now, we're growing the COR resistant cultivar Maite as well as Fukunaga for the nematode resistant rootstock. We use these bioreactor to multiply them. And right now we have 5 to 700 plantlet about this size in the lab right now. At one point, we had thousands, but we had two massive contamination events and we lost them all. It was a very sad day when we had to autoclave everything. We did have to professionally clean our air duct and clean everything to get everything under control. And these are all the people that's involved, probably not all. I probably forget some of the people, including the collaborators lab and the team at HARC. Okay, this is all I'm going to say today. Any questions?